This is a GCSE presentation looking at the uses of monoclonal antibodies. To get started, describe the 6 main stages involved in the production of monoclonal antibodies. Pause the presentation, and when you're ready you can restart. So the first stage is that a vaccine has to be injected, or anything that contains an antigen, but normally it is a vaccine. The mouse responds, and when its immune system produces lymphocytes, remember lymphocytes are white blood cells, those lymphocytes will produce antibodies, and those antibodies are specific and complementary to the antigen that we've injected. We then have the spleen cells removed, and these spleen cells will be producing the lymphocytes. We then take those cells and we fuse them with a cancerous white blood cell, which is known as a melanoma. And when we combine a melanoma with the spleen cells, we produce what is called as a hybridoma. A hybridoma is a cell that will continually divide, and every one of those cells is identical, and every one of those cells will produce large numbers of monoclonal antibodies. And all of those monoclonal antibodies, because the cells are clones, will be specific and complementary to the original antigen. The last thing we do is we collect up those antibodies and we purify them. In this lesson, we're going to look at some of the uses of monoclonal antibodies and explain how the different processes work. So monoclonal antibodies, the term comes from the fact that all the antibodies that are produced by any one particular B cell are identical copies of each other, and they are clones. And mono simply comes from the fact that it's one type of antibody. So we have one type of antibody, but we have lots and lots of copies of it, hence we use the term mono and clonal. Each monoclonal antibody is specific to a particular antigen and is also complementary, so not identically shaped, but the shapes fit together like two jigsaw pieces perfectly. Now, when we first produced monoclonal antibodies, it was believed that it was going to be the kind of silver bullet in terms of treating diseases and infections. Unfortunately, we have found that using monoclonal antibodies, there are a number of side effects, and as a consequence, it has not been as widely used as we first thought. And there's still a lot of research going on, but it certainly was not the silver bullet that everyone thought it was going to be. So antibodies in your immune system play two specific roles in terms of destruction of a pathogen. Look at the two pictures below, see if you can work out what the job of the antibodies is in terms of the immune system. Pause the presentation, come up with your best answer, and when you're ready you can restart the presentation. So pause now. Well, the two pictures, the bottom left, we have large numbers of pathogen being held together by antibodies, and we refer to that as agglutination. Now, GCSE can use the word clumping, but agglutination is the kind of the scientific, more accepted terminology, particularly if you're going into medicine and that kind of thing. What happens is that basically the bacteria cells or the pathogens are clumped together into large bundles, and it makes it much easier for a certain type of white blood cell known as a phagocyte to engulf and digest that particular lump of pathogens. The second job of an antibody is antibodies can act as markers to get those pathogens to engulf bacteria cells or engulf pathogens. So they will actually stimulate phagocytes, and they will encourage phagocytes to move wherever they are in order to target specific pathogens. So antigens and antibodies are referred to as being complementary. What that means is that they're not the same shape, but they fit together in the same way the two jigsaw pieces fit together. If you look on the screen, you've got 3 antibodies and you've got 3 pathogens. Each pathogen has a different antigen on its surface. All you have to do is match up the antibody that would go to each of the different pathogens. Pause the presentation now, work out which one goes where, and when you're ready, restart the presentation. So antibody A would target the middle of the pathogens. Antibody B will target the one on the right, and antibody C will target the one on the left. Notice that A and B will have no impact on that pathogen other than the one in the middle and on the right hand side, because they are not complementary shapes to the pathogen that is targeted by antibody C. When we're talking about uses of antibiotics, there are lots and lots of examples that they can give you in the exam. There's no specific examples that you have to know, but there are a few suggested examples that you should look at. The most important thing is to remember that the idea that they're complementary is how any process that involves the use of antibodies works. The antibody has to be complementary to whatever it is you're looking to target, whatever it is you're trying to find. There are three main uses. One is to target drugs to specific cells, for example, targeting cancer cells. We've got medical diagnosis, and we've also got the traditional example, which is pregnancy testing. On the right of your screen, there are two images explaining how drugs can be targeted to specific cells and how we can use antibodies to stop cancer cells. We've got direct monoclonal antibody therapy and indirect monoclonal antibody therapy. Use the pictures on the right hand side and try and come up with a couple of sentences explaining how the antibody is stopping the growth of the cancer cell. Pause the presentation now, and whenever you've got your answers, restart and check. In the first picture, we have a chemical signal that goes to the cancer cell to tell that cell to multiply. Remember, cancer cells are cells that multiply uncontrollably. They're continually doing the process of mitosis and making new cells that are also cancer cells and are identical genetically. If we can use an antibody to block the receptor that the chemical signal is binding to, then the chemical signal no longer tells the cancer cell to divide and the cancer cell stops. And that's what happens in direct monoclonal antibody therapy. In indirect monoclonal antibody therapy, what we do is we use the antibody to deliver a particular substance to a cancer cell known as a cytotoxin. We can use it to deliver other drugs as well, but in this particular example it's cytotoxin. In an exam, if they change the name of the substance, simply refer to whatever the antibody is delivering. But normally, whatever substance is delivered, it will actually kill the cancer cell. And because the antibody is complementary to the antigens only found on the cancer cell, none of the normal body cells are affected. So we have less side effects. We can also use monoclonal antibodies for medical diagnosis or identification of any substance using fluorescent markers. What we do is we get different antibodies that we know attach to different substances. So we have to know in advance which antibody will attach to which substance. We then put a fluorescent marker of different color onto all the different types of antibody, and we then add them to a sample of a patient's blood. We leave it for a bit of time, and then when we wash away, what will happen is that if there is a complementary antibody, an antigen, they will bind, and that fluorescent marker will stay on the slide, whereas all the other fluorescent markers will be washed off whenever we wash the sample. When we then look at the color of the slide, we'll be able to see which antibody has stuck, A, B, or C, and as a consequence we can use that to identify what type of disease, protein, or substance is present in the person's blood. In this particular case, which of the antibodies is going to attach to that particular protein, A, B, or C? Well, the correct answer is C, because the complementary antigen antibody shapes fit together. A and B are not complementary shapes, so they would not bind to that particular protein. So when the slide was washed, they'd simply be washed away. And when you looked at the slide at the end, you'd see the color C, which is teal. Now there are some advantages for using this particular method. It means that we can very quickly do large numbers of samples, and it can be all automated in machines. And what we can also do is we can use the machines to measure wavelengths of light, and it takes away that human element, because quite often misdiagnosis occurs because of the human element. If we can get machines to read specific values, we are much less likely to get false negatives and false positives. Pregnancy testing. Pregnancy testing is based around a simple interaction between antibodies that are complementary to specific antigens. When you have a pregnancy test, you've got three specific areas. You've got a reaction site, a test site, and a control site. At the reaction site, we have antibodies that are attached to enzymes, which are the orange circles, and they are free to move along the paper. So if the paper gets wet, the water will move along, just like if you get kitchen roll wet, the water will soak up, and it will carry that antibody along this test strip. At test site 1, we have an antibody, and it is stuck there. It cannot move. Antibody B cannot move from that location. And antibody B also has some substrate, and that substrate will change color if the enzyme is there long enough. The third area is the control site, where we have antibody C, and again antibody C is fixed, it cannot move. There is also some substrate there, which again, if the enzyme is next to that substrate long enough, it will cause a color change. And what we're basically looking for is a substance called HCG, and HCG is produced and found in urine of pregnant women. Let's have a look at how it actually works. What we've got here is what happens during a negative test. Now, the first time you watch this, I would suggest that you watch it and I'll give you the commentary as we go through step by step. However, just to test your knowledge and understanding, you may want to go back and replay it, but replay it with no sound so you don't get to hear my voice. And you should then be able to explain what's happening at each step. And if you pause it, you'll be able to see it and work your way through and hopefully get the right sequence. But for now, I'm going to explain. So the first thing that happens is we have to have a urine sample added onto the test strip. If a urine sample is added onto the test strip and we're getting a negative test, it means there is no substrate that will attach to antibody A. There's nothing to attach to antibody A, so we have antibody A left on its own. Because the test strip is now wet, the antibody A will then move along from the reaction site into the test site. At the test site, nothing happens because there is no complementary shapes involved. So what happens next is the antibody moves along and goes straight onto the control site. At the control site, we do have a complementary shape, so antibody A will attach to the antibody that is fixed in position on the control site. And as a consequence, we will have a change in the substrate. With the change in the substrate, we have a change in color, and we get a positive line on a test strip. With only one line on the test strip on the control site and nothing on the test site, we would say that this is a negative test. In a positive test, we have a different thing occurring at the test site. Again, we need a urine sample. When we have a urine sample, if this person is pregnant, they will then have the substrate, and as a consequence at the reaction site, we now have some antibody with enzyme attached but no substrate, and some antibody with enzyme attached and the substrate attached. That then proceeds into the test site because again the urine's made the test strip wet, so it'll just pass along. This time round we have some of antibody A with no substrate attached and it'll sit there and do nothing, whereas the substrate that is attached to antibody A is also complementary to the antibody at the test site. That means some of antibodies A are stuck at the test site, which means that the enzyme is stuck at the test site. We continue along, but when we have enzyme stuck at the test site, we will have the substrate change color. as the substrate has changed color, we now have a positive line on the test site. The urine continues to move along, and the antibody A, which has no substrate attached, will attach to the the antibody that is fixed at the control site. Remember, that antibody is also connected to some substrate, and because antibody A and the enzyme are now attached at the control site, we will also get a color change at the control site, so we will have another blue strip appear. And now we can see on a positive pregnancy test, we have 2 blue strips. As a result, this person is going to have a baby. There are some ethical issues around the use of monoclonal antibodies that you should be aware of. We are experimenting with animals. Now, yes, we can argue that it's not cosmetic, it's medical treatment, it's saving lives, it's important, but you are still taking animal life in favor of human life. We've also known that some of these treatments have been linked to deaths. Some of these treatments, the thought was that monoclonal antibodies, when your body makes antibodies, these will be fine, these will be brilliant, nothing's going to go wrong, but actually quite a number of studies have found quite significant side effects from these monoclonal antibodies, and we're still refining the technique. There's also an issue in terms of actual trials, because while the safety test stuff on animals, when you start testing it on humans, it seems to have very, very different effects. Now, that can also be true of conventional drugs, but it has been noted that the variation between conventional drugs and monoclonal antibodies is quite significant. So again, it's still being refined. It's still a pretty modern process, and we're still working on it, but there are some issues. Last thing to finish off, do the benefits of monoclonal antibodies outweigh the costs of animal life? There's no right or wrong answer, folks, just whatever your opinion is. It'd be even better if you can go and research some papers or some journal articles or some books and provide some references whenever you're actually putting your argument forward, and make sure that you've got a good solid argument.